Hello, I'm Steve Kelly, President and CEO of Carisma Therapeutics. I want to thank the organizers at the Alliance for Regenerative Medicine for inviting us to participate. I'm going to spend the next few minutes talking about Carisma and our overall engineered macrophage platform. Carisma is a pen spinout that was formed in 2016 by Sar Gill and Mike Klichinsky. They saw the challenges that lymphocytes are seeing in the treatment of solid tumors, and they wanted to approach it with a different effector cell, the macrophage. The difficulty there is that macrophages are resistant to traditional vectors like lenti and AAV. And it was only after they identified Ad5F35 as a good vector that had excellent transduction efficiency that they could move forward. Today, we have a pipeline of 3 agents. Our lead program is in a phase 1 study at Penn, UNC, and the City of Hope, the HER2-targeted CAR macrophage. We also have a mesothelin-targeted CAR macrophage and a PSMA-targeted CAR macrophage in development. We also have a very active discovery group that's leveraging our increasing knowledge of macrophage biology and how to tune those macrophages, and we have a growing pipeline as well. As you know, manufacturing is a critical element of cell therapy approaches, and so we've invested a lot of time, effort, and working with our partners, WuXi and Miltenyi, on vector and cell product respectively. We've run multiple donors through our process, and we're very happy with the approach so far. Our team has continued to grow. We're currently 50 employees, all functions represented within the company. We also have a great network of advisors that include Sar Gill and Carl June from Penn, but we also have Hai Levitsky, former CSO of Juno. We have macrophage experts like Lisa Cousins out of OHSU. We have solid tumor KOLs like Prasada Samili out of Memorial Sloan Kettering. It's a great group of advisors helping us realize the potential of our platform. To date, we've raised around $120 million in 2 financing, 60 in 2018, another 60 earlier this year. It gives us the cash runway to hit key development milestones and that we'll talk about in just a moment. Now, why are we so excited about macrophages and the approach? And it comes down to the challenges that people experience with cell therapy overall in solid tumors. And it comes down to tumor access, an immunosuppressive tumor microenvironment, and antigen heterogeneity in solid tumors. Macrophages are ideally suited to address each of these challenges. Macrophages are recruited to the tumor along a chemokine gradient. If you were to look at any tumor microenvironment, you're going to find more macrophages than any other immune effector cell. We're going to leverage that natural biology of getting into the tumor. Once our cells arrive, they're going to engage antigen, and they're going to start to phagocytose or eat the cancer cells. And they'll continue to eat for the life of the macrophage, which we think in humans will be 3 to 5 months. As they're eating, they're going to be producing a variety of cytokines and chemokines that have a net effect of warming up the tumor microenvironment and recruiting in other effector cells. We see an influx of T cells, NK cells, dendritic cells. We see conversion of existing TAMs into an M1 phenotype. Lastly, and importantly, macrophages are one of the 3 professional antigen presenting cells. And as they're eating through the tumor, they're going to be processing and presenting various secondary antigens out through MHC to the T cells that are recruited in. The goal, of course, is epitope spread and establishing long-term immune memory. So what I like to say is that we are mimicking the innate immune system and driving true adaptive immunity. And the hallmarks of our approach are 1, the ability to traffic to tumors, 2, the ability to kill directly through phagocytosis, 3, the ability to warm the TME and recruit in other effector cells, and 4, the ability to generate an adaptive immune response. So to go after this, of course, we need to develop an engineering platform. And so there's multiple parameters here. 1 is the ability to engineer the cells. As I said, Ad5F35 was really the seminal moment to move forward with macrophages. But since then, we've also been able to identify and license the only other viral vector we know of that works in macrophages, VPX lenti. We licensed that from NYU. And lastly, we have modified mRNA that we've been able to use. It gives a transient CAR expression, but still it's a useful tool for us. As I said, we were able to engineer macrophages. More recently, we've been able to engineer monocytes, which could be considered a precursor to macrophages, and we're doing development work there. Lastly, we have a variety of payloads that we can produce. They include CARs, of course, but anything related to that, other cell therapy approaches, logic gates, etc. But we can also add other transgenes, such as things for cytokines or antibodies or bispecifics, things like that. And we've also been able to engineer, or I should say, we've been able to mobilize CRISPR and do gene editing on macrophages as well. So we have a very robust platform to build on. which is what we've been doing with our current pipeline. As I said, CT-0508 is a HER2-targeted CAR macrophage for applications in HER2-positive solid tumors. IV route of administration, we're currently in a phase 1 study. We're also leveraging that same product for regional administration. There are some case examples where we could look at regional administration in ovarian cancer, intraperitoneal, for example, or we could look at CNS administration for HER2-positive brain malignancies or HER2-positive brain mets. We've also been doing a lot of work on what's the ideal regimen, CT-0508 alone, or could we enhance the activity with some work in combination with anti-PD-1 or anti-CD47, and so we're doing work there. As I said, we have 2 other programs in the pipeline, but we also have a very active R&D engine that's looking at allogeneic CAR macrophages, heme malignancy, neurodegeneration, and we do have some effort in liver fibrosis as well. Now, we've published a lot of this work in the past showing that we can do all the things that I've talked about, phagocytosis, killing, cytokine production, etc. I do want to spend just a couple of moments and talk about a fully immune, competent mouse where we're able to see mobilization of that adaptive immune response. So here there's a mouse with an intact immune system. We have implanted a CT26 HER2-positive colon carcinoma and treated the mouse with our CAR macrophages. And you can see in the graph in the middle, good responses. In this case, it was 5 out of 8 mice had a response. And if you look at the survival curves, you can see that that's durable out through, in this case, 75 days when we stop looking. Importantly, on the lower half of the slide, you can see that we're looking for T-cell infiltration, and control and untransduced macrophages have little infiltration, but the CAR macrophages, you can see in brown, a significant CD8 count coming in, and it's also demonstrated on the right-hand side in those graphs. That's important because this signals that we're able to bring in those T cells and hopefully present antigen and generate an adaptive immune response. Now, that's what we wanted to see. So we actually implanted 2 different tumors in one mouse, a dual flank model, where we have a HER2-positive tumor on one side and a HER2-negative tumor on the other side. In all the respects, they're identical. In this case, we treated the mice with the CAR macrophages. We were able to see, as you see on the lower left, good response. 6 out of 8 mice had a response to our CAR macrophages. And importantly, in the graph there in the middle, on the HER2-negative tumor, we had an abscopal effect. We saw, as compared to untreated and untransduced macrophages, a reduction in the growth rate of the tumor. Now, we know our macrophages will have no effect on HER2-negative cells, so this has to be driven by another approach. And so we pulled those tumors and took a look at what's happening in the TME. As you can see in the graphs on the right, we see a good influx of T cells, both CD8s and CD4s. We see some specific CD8s against a known class 1 antigen in this tumor model, GP70. So evidence of epitope spread. We see NK cells come in, dendritic cells come in. So we are mobilizing the immune system and generating an abscopal effect. This doesn't tell us what is really driving the outcomes in the mice. Is it the CAR macrophages, or is it the T cells? Nor does it really reflect actually what's going to happen. So what we want to do is look at another model, and it's a Rechallenge experiment. So our goal, of course, is to kill directly through phagocytosis, generate epitope spread through the T cells, and maintain that through long-term immune memory. So we took the mouse model again and planted our CT26 HER2-positive tumor, treated with our macrophages, and took the complete responders and set them aside for a couple of months. Then we rechallenged with a CT26 wild type. Again, our macrophages will have no effect. So anything that is presented has to be T-cell mediated. And we compared that with naive mice. As you can see in the survival curve on the right, all of the naive mice succumbed to disease within 35 days, but none of the previously treated mice did, and they all resisted tumor growth. And so this is really the first time this is indicative of being able to generate that T-cell response. And we've repeated the experiment for longer and knocked out memory T-cells, and we know that it's a T-cell mediated approach. So we're really excited. And as I said, we've moved into the clinic. Now, I'll talk briefly about our lead program, CT-0508. It's, again, a HER2-targeted CAR macrophage. The CAR construct is shown on the upper right, an SCFV derived from Herceptin, CD8 hinge, CD8 transmembrane, and a single signaling domain, CD3 zeta, which we've found to be the most potent phagocytic signal we can put into a macrophage. We currently have a phase 1 study ongoing, 3 sites today, Penn, UNC, and City of Hope. 3 patients enrolled to date. And by the time you're seeing this, 3 patients treated to date. Our goal is to continue to expand the number of sites, getting up to at least 3 additional sites to increase the pace of enrollment. And our goal is up to 2 patients a month for a total of 18 patients. Our plan is to present data at SITC coming up very shortly, and expand a data set at ASCO next year. In addition to this initial clinical program, as I said, we're working on regional administration and a T-cell checkpoint combination. One last point is that just this month we received FDA Fast Track designation. Now, as I said, manufacturing is an important element of cell therapy, and we've spent quite a bit of time optimizing our approach. We have currently a 7-day process where we take patients apheresis. We pre-mobilize patients with GCSF, have them sit for apheresis, collect the leukopak, and isolate CD14 positive monocytes. We take those monocytes, differentiate into macrophages, and then transduce on day 5, harvest and crop preserve on day 7. Our yield is around 1 to 5 billion CAR macrophages, and our vein-to-vein time is approximately 26 days. Now, our clinical study is shown on this slide. It's a basket design, phase 1, multi-center study. And the goal is to establish safety and feasibility of using CAR macrophages in an autologous approach. We're going to be enrolling 18 patients in total, HER2 overexpressors, 2+ and 3+. And as I said, we have 3 patients on study already. The initial group of patients, or 9 patients, will have a safety run-in where we're looking at fractionating their dose. So on days 1, 3, and 5, they'll receive 500 million cells, 1.5 billion cells, and up to 3 billion cells, respectively. In group 2, not only are we going to be bolus administering the full manufactured dose, we're also going to be radiolabeling with zirconium-89 and imaging with PET to show localization of our macrophages in tumor. As I said, the primary objectives are safety and manufacturing feasibility, but we also want to show a number of other clinical correlates of activity. So we have 2 post-treatment biopsies scheduled, 1 at 1 week, 1 at 4 weeks. And we'll look at things as shown on this next slide. So we're going to be looking at the PK, so the rapid transition from circulation into tissue. We'll look at the mechanism of action. So we're looking at cell killing and TME activation. And we'll also be looking at adaptive immune response. And this is all data that we plan to present as we go forward. Now, I also mentioned our platform, and we're doing quite a bit to enhance the overall activity. Our phase 1 study will establish a baseline of activity for a CAR macrophage approach. But we're looking at ways to enhance that activity. So combination approaches. We've seen preclinically there's synergy with a combination with a T-cell checkpoint inhibitor. So we're working on that. We've seen combination with anti-CD47 antibodies have some enhanced activity. So we're working on a study there. So there's really a lot of optimization on how to best use our CAR macrophages. We've also enabled CRISPR editing, so we can do things like knock out SIRP alpha. So block that CD47 SIRP alpha axis so that don't eat me signal. So we've been able to enable that. We've also made a number of novel CAR designs. We made over 200 different constructs, and each element is important: binding efficiency, hinge length, the signaling domains. And so we think that there are ways that we can enhance the activity there. And lastly, we're looking at multiplex engineering, putting in other genes so we can do things like produce a variety of cytokines in tumor to enhance our activity. In addition to the work we're doing on the macrophage itself, we're also looking at ways to enhance our manufacturing process. I mentioned CAR monocytes. CAR monocytes are a way to produce probably double the number of cells on an autologous approach, and may provide a trafficking advantage where it remains to be seen, but there are smaller spherical cells, and so we may be able to get more cells into tumor, which we believe will translate into better activity. Lastly, and importantly, we're working on allogeneic CAR macrophages, working with the Blazer Lab at Minnesota. We've demonstrated that we can make allogeneic macrophages and we can make allogeneic CAR macrophages in two different approaches. So we know that we can express CAR, we know they're polarized towards an M1 phenotype, and we're doing functional testing now. So we're really excited about the eventual usage in an allogeneic approach. Now, just in terms of this is the team, I'll jump through that. But in terms of the upcoming milestones, as I said, our third patient will be dosed by the time you're seeing this presentation. We expect to enroll the study completely by the middle of next year. We'll have data presentations at SITC in November of this year, as well as ASCO in June of next year. Our lead program for mesothelin has been identified. We're moving that into IND-enabling work with a planned IND filing approximately a year from now. PSMA the lead will be identified at the end of Q1, and a similar path to IND filing. We have ongoing work. I mentioned a couple of things in the platform enhancements, the combination approaches, gene editing, et cetera. We have our CMC enhancements that we're working on. All that will be ongoing and will be incorporated into subsequent phase 2 development toward the latter half of next year. So a lot of activity over the next 12 to 18 months. So just to summarize, Carisma really is the leader in engineering macrophages. We have a proprietary platform, good IP supporting it. We have an emerging pipeline of different agents. We have a GMP supply chain. We're using CDMOs. We have an experienced team and a great team of advisors and multiple value catalysts over the next 12 months or so. I want to thank you for your time, and it's been a pleasure, and I look forward to catching up with everyone in the near future.